जी बॉटनी भूतपूर्व लेक्चरर एंड ट्रांसफर आपका ये मथुरा. Nowadays I am teaching to the students of CSC board, UP board and BSc, MSc and IAS PCS students. Now I am teaching you double fertilization. In double fertilization takes place in ovary. First male gamete combined with egg cell and zygote is formed. Then second phase is called triple fusion or triconsanguine. द्वितीय क्रोमोसोम या second male gamete plus secondary nucleus is equal to endosperm mother nucleus or PEN, primary endosperm nucleus. Now we see the different diagram how these enter into the fertilization. First antipodal cells, then pollen nuclei, then synergid, then pollen tube. It enters into the micropyle and leaves to the male gamete into the ovule inside, and it combines to the female gamete. In the second diagram we see the first male gamete, then female gamete, both combines, and male and female gamete combine to each other. कहलाजा डिजेनरेटिंग एंटीपोडल सेल्स खत्म हो जाती हैं. प्राइमरी एंडोस्पर्म न्यूक्लियस या पेन, PN पेन, एंडोस्पर्म मदर न्यूक्लियस शेष रहती है. प्लांट फर्टिलाइजेशन स्ट्रक्चर एंड इवेंट्स. embryo development and dicotyledonous plants. First of the zygote, it forms a transverse division, suspension cell and embryonic cell is formed. Then two cells, then a long suspension is formed of different cells, and the first cell is called hostorial cell. Hostorial cell provides the food. Now we see in the dermatogen, then perivallum, then plerome. Suspension is long structure and two structures are called cotyledon, that is formed from the embryonal cells. Cotyledons from the suspension. If you see in the diagram, we can see inner integument, suspension, outer integument, endosperm mother cell, nucellus, food region, stem region, then cotyledon. If we take in monocot stem, the first structure is called oospore, that is formed by the formation of male and female gamete. Then transverse division takes place. Upper first cell and lower terminal cell is formed. Then proembryo is formed. This proembryo formed from suspensor and embryonal cells. Then a structure is formed. Then a hypocotyle, epicotyle, and plumule are formed in monocot stems. In monocot stems, plumule is formed, cotyledon is formed, hypocotyle is formed, radical root cap is formed, and in the embryo, cotyledon, coleoptile, and shoot region, and epiblast and radicle is formed. Now we proceed that types of endosperm. Endosperm is that structure which provides food material to the embryo. Types of endosperm are following types. First is nuclear endosperm. Nuclear endosperm में pollen tube you can see, then zygote is formed and primary endosperm of the nucleus and antipodal cells are present. Next, zygote, endosperm and antipodal cells are formed. Then zygote, endosperm and antipodal cells are formed. Then proembryo and central vacuole is in the center and free nuclei are formed. It is called free nuclear endosperm. Next is cellular endosperm. Cellular endosperm, in this first pollen tube, then zygote, then endosperm nucleus, then central endosperm, then cell ostronium. In this division, whorls are formed in each cell, and a endosperm is formed that is called cellular endosperm. Next, cellular endosperm. It takes place in the polypetally, and coconut water is the example of this. White kernel is present. And the heliobel endosperm, it is found in Aeromurus. In Aeromurus, you can see in endosperm, this is formed by two cells. First, transverse division, cell wall. Then various nuclei are formed. You can see that the free nuclear division are formed, and on the upper side there is formed cellular division. When the cellular division is a connecting link between heliobel endosperm is connecting link between cellular endosperm and heliobel endosperm is formed. It contains cellular endosperm and free nuclear division both. Now histology of endosperm. After fertilization, the embryo contains ovule, hilum, hyphal, fundal, obturator foramen, exostroma, endostoma, outer segment, inner segment, nucleus, endosperm and embryo are formed. We can see development of seed and formation of fruit. First we see in a फ्लावर फर्स्ट कैलिक्स गिर जाते हैं, कई फल के गिरते हैं, कोरोला प्राय गिर जाते हैं, स्टेमन गिर जाते हैं, कोटोलिडोन होते हैं, स्टिग्मा गिर जाता है, स्टेल गिर जाता है, ओवरी फ्रूट में बदल जाता है और ओवरी पेरिकार्प में बदल जाती है और ओव्यूल सीड में बदल जाता है. वी कैन सी इन ए सीड it is micropyle, then seed coat, then endosperm, then cotyledon. In this fruit, shoot is special and meristem and shoot tip is found, and a small structure is called corauncle. The cotyledon contains root, meristem, hypocotyle and root axis. In the major stem, if you see the diagram, a layer is found that is called epithelial lining. On the upper side it is found endosperm, and on the lower side is found embryo. It contains cotyledon that is called scutellum, and radicle and coleoptile and coleoids are present on the above part. In apple it is a
8 मिनट हो गए.
हैं?
8 मिनट हो गए. 8 बोल लो और ये पे बोल लो.
अच्छा. In the false fruit we can see that the thalamus form the fruit formation, and the seed is present, endocarp is present, mesocarp is present, and in strawberry you can see thalamus and achenes. बस यहाँ तक.